Okay. So, hi, today is the last problem solving session of this course, Overview and Integration of Cellular Metabolism. Uh, I hope my voice is clear. If you can confirm once.
Yes, ma'am, it's clear, ma'am.
Okay. Okay, then so as you know, the course instructors are Professor Aditri Beer and Professor Arindam Ghosh from IIT Kharagpur. I'm Shrija Ghosh of PMRF from IIT Kanpur. And today we'll discuss the last topics that are left for this course. We'll be discussing metabolic control analysis, and then we'll discuss fatty liver and alcohol metabolism and aspects related to obesity. So, I have prepared certain questions related to all those topics, and I'll be discussing the questions. Uh, Along with that, if you have any doubt in any of the questions or in any of the topics that are there in this week, do let me know. I'll try to help you out. Okay. So I'll just start in a minute. Yeah. So starting with the first question, flux control coefficient in metabolic control analysis measures the sensitivity of flux to changes in what parameter? Option A, substrate concentration. Option B, enzyme concentration. Option C, temperature. And option D, coenzyme availability. Flux control coefficient in metabolic control analysis measures the sensitivity of flux to changes in which parameter? Option A, substrate concentration. Option B, enzyme concentration. Option C, temperature. Option D, coenzyme availability. You're saying option B, that is enzyme concentration. That is the correct answer. So, flux control coefficient, basically, it is the measure of the relative contribution of each enzyme to setting the rate at which the metabolites flow through the pathway. So we have a pathway here. So you can see here is a hypothetical pathway. X goes to Y, goes to Z. So like any metabolic pathway. So we have studied so many metabolic pathways in the previous session. We have studied uh, glycolysis, TCA, all those pathways, right? So uh, now you see every enzyme has a substrate and it makes one product. This product is again a substrate for another enzyme, and this is a cascade reaction. Okay, this is a flow of like series of different enzymatic reactions happening. Now, flux control coefficient basically is determined not by one enzyme but all these enzymes together. Okay, so concentration of one enzyme can actually influence the concentration of other two enzymes. So if the concentration of this one enzyme goes up, let's say, then what will happen? Uh, since the amount of enzyme is higher, more substrate will be used up. Eventually, Y will go down. And since Y concentration of Y goes down, what will happen? Activity of Xa's will increase. Why? Because now the concentration of Y is going down. That means the pathway will sense that we need more and more Y to meet the requirement by YDH, that is higher. Okay. So what the pathway will do? Pathway will convert more X0 into Y so that more Y can be catalyzed by the enzyme YDH. And when YDH will go up, obviously concentration of Z will also go up, and concentration of Z being higher means the activity of ZAs will also be higher. So you see, concentration of one enzyme is influencing actually the whole metabolic pathway. And this flux control coefficient of an enzyme, it is a system property. It is not related to one enzyme in isolation. It is related to all the enzymes in the whole pathway. Okay, so that is the major thing. And it can have values from 0 to 1. Uh, and It also depends on the concentration of substrates and the external metabolites. So this flux control coefficient, it can be negative as well, because if there's a branching in the pathway, let's say uh, X goes to Y goes to Z, this is one pathway. Okay. Now there's one more pathway branching here where Y is making M and N. Okay. So now, and let's say the enzyme here is A, enzyme here is B, this is C, and this is D. Okay, so now if the enzyme concentration C increases, that means automatically this pathway, the pathway that is making Z, this pathway will be down-regulated because all the substrate Y that is required for making Z is diverted towards another pathway that is going towards M and N. Okay, so in that case, enzyme concentration of C, basically the enzyme C will have a negative flux control coefficient on the pathway that is making Z from X. Okay, so that is how flux control coefficient can be negative as well. But mainly for one single, when we are talking about one single pathway, the value ranges from 0 to 1. Okay, I hope that's clear. Moving on to the next question. Which coefficient in metabolic control analysis quantifies the responsiveness of an enzyme to metabolite concentration changes? Which coefficient in metabolic control analysis quantifies the responsiveness of an enzyme to metabolite concentration changes? Option A, flux control coefficient. Option B, elasticity coefficient. Option C, response coefficient. And option D, rate constant. Which coefficient in metabolic control analysis quantifies the responsiveness of an enzyme to metabolite concentration changes? Option A, flux control coefficient. Option B, option B, elasticity coefficient. Option C, response coefficient. And option D, rate constant. Uh, you have answered option A, right? That is flux control coefficient. Actually, the correct answer is option B, that is elasticity coefficient. So I just now told you that flux control coefficient is basically uh, is a it is dependent on the enzyme concentration in a metabolic pathway. So it basically determines how fast or slow the whole pathway can move. Okay. But elasticity coefficient actually gives the responsiveness of the enzyme to particular metabolite concentration changes. So the concentration of each metabolite or an activator or an inhibitor, how that is affecting the whole pathway, that is the elasticity coefficient. So elasticity coefficient is actually given for each enzyme individually. Like I told you, flux control coefficient is not given for each enzyme individually. It's actually a integrated thing. Whereas elasticity coefficient, it is a quantitative expression of the responsiveness of a single enzyme to changes in the concentration of the metabolite or regulator. So basically, it can have positive values for the metabolites that stimulate the reaction. So for a enzyme catalyzed reaction, A goes to B. So if we have a reaction A goes to B, catalyzed by the enzyme E. So obviously, if more A is present, that means there will be more substrate available for the enzyme and the reaction will go in the forward direction. That means substrate is will have a positive effect on the enzyme's activity. Uh, so the concentration of this substrate, that is one of the metabolite, it is how it is affecting the enzyme's activity, enzyme's kinetic property. Okay. So elasticity coefficient is positive for the metabolites that stimulate the rate of reaction and negative for the metabolites that slow the rate of reaction. So if the concentration of B goes up, that means the pathway will get the signal, okay, we don't need more B. So the enzyme E will be negatively regulated. Basically, enzyme E won't be making more of the product B, so that uh, elasticity coefficient for this enzyme will be in negative. That means now the uh, reaction has slowed down. Okay, is this clear to you?
Yes, ma'am. But even flux control, um, it is a responsiveness of a single enzyme they have mentioned, no, ma'am. The effect of a
No, no,
single
no.
enzyme on the metabolic pathway. How to differentiate these two, ma'am? Flow
So
control and elasticity.
this is the major difference, that elasticity coefficient depends on single enzyme, uh, like responsiveness of the single enzyme. Okay, whereas flux control relative contribution of each enzyme. Okay, so when we are talking about flux control, if we are looking on the enzyme YDH here, the if contribution of YDH will actually influence the activity of XAs and ZAs as well, right? So that is the flux control. Flux control coefficient will depend on the whole pathway. It is not for one single enzyme. It is for the whole pathway, that how fast the pathway is moving or how slow the pathway is moving. Uh, is this clear to you now?
Okay, ma'am.
So basically, flux control coefficient, when we are talking about flux control coefficient, it is showing how slow or how fast the whole pathway is moving. Okay. So let's say we are talking about glycolysis. So if I'm talking about the flux control coefficient, in case of glycolysis, it will measure the sensitivity of the whole pathways flux, basically how fast the final product, that is pyruvate, is forming, or how slow so if already a lot of pyruvate is present, we have studied about feedback inhibition, that if more and more product is present, then the pathway will be negatively regulated, and uh, the overall speed of the pathway will be slowed down. So flux control coefficient, in simple words, you can say that it is kind of giving a quantitative term to the speed of the reaction, speed of the whole pathway. Okay. Whereas when we are talking about elasticity coefficient, it is kind of a local property. It is measuring the sensitivity of one enzyme's rate, how one enzyme's rate is affecting the pathway. Okay. So if I'm saying that this particular enzyme, uh, like if I'm talking about the concentration of Y in this pathway. So for flux control coefficient, I was talking about the concentration of the enzyme YDH, right? So the concentration of enzyme YDH was affecting the speed of the pathway, or concentration, let's say concentration of concentration of Xa's will be down, amount of Y will be less, amount of Y will be less, then amount of Z will be less. But then all these things are dependent on the enzyme concentration. Whereas when we are talking about elasticity coefficient, we are talking about the metabolite concentration, not the enzymes concentration. We are talking about the metabolite concentration. Like now we can say that if the concentration of Y is less, uh, in that case, the enzyme's activity will be slower because there is not a lot of substrate for the enzyme to catalyze. So the reaction will proceed slower, slowing down the overall rate of the pathway. But here we are talking about the metabolites that are affecting the enzymes. Okay. For flux control coefficient, we are talking about the enzyme concentration, whereas when we are talking about elasticity coefficient, we are actually talking about the metabolites concentration. So either the activators or the inhibitors. So we have studied about different allosteric inhibitors also, right? Or allosteric activators. So those concentration of all those metabolites will also come under elasticity coefficient. Is this better now?
Yes, ma'am.
So moving on to the next question. In a multi-enzyme pathway, the sum of all flux control coefficients equals option A 1, option B 0, option C 100, and option D, it depends on the pathway. In a multi-enzyme pathway, the sum of all flux control coefficient equals option A 1, option B 0, option C 100, and option D, it depends on the pathway. You're saying option A, that is 1, that is the correct answer. So this I've already mentioned that the flux control coefficient ranges from 0 to 1. So 1 means the flux, basically the speed will be at the maximum rate. Okay, and 0 means there's no flux at all. The next question, metabolic control analysis primarily studies option A, structure of enzyme, option B, how enzyme activity affects overall pathway flux, option C, hormonal signaling pathways, option D, gene expression of enzymes. Metabolic control analysis primarily studies option A, structure of enzyme, option B, how enzyme activity affects overall pathway flux, option C, hormonal signaling pathways, and option D, gene expression of enzymes. Gene expression of enzymes. You are saying option B, that is the correct answer. So, metabolic control analysis is the analysis where we study how the enzyme activity affects the overall pathway flux. So, basically, now we are looking at the uh, overall pathway, how it is being regulated. So we have studied about the regulation of different pathways, right? So metabolic control analysis, the parameters that are there in the metabolic control analysis, basically the flux control coefficient, and then elasticity coefficient, and then uh, so all these parameters, these are basically designed, or these are basically mathematical terms and functions, and these are defined to define the whole regulation of a pathway. in terms of the enzyme's activity, in terms of the enzyme concentration, in terms of the metabolite concentration. So these are mathematical terminologies that are defined. Okay. Question number 5. Adiponectin and adipokine exerts its major metabolic effects through activation of which enzyme? Adiponectin, which is an adipokine, exerts its major metabolic effects through activation of which enzyme? Option A, glycogen synthase. Option B, pyruvate dehydrogenase. Option C, AMP activated protein kinase. And option D, glutamate dehydrogenase. Adiponectin, which is an adipokine, exerts its major metabolic effects through activation of which enzyme? Option A, glycogen synthase. Option B, pyruvate dehydrogenase. Option C, AMP activated protein kinase. And option D, glutamate dehydrogenase. You have answered option C, that is AMP activated protein kinase. That is the correct answer. So adiponectin, it is a peptide hormone. It is comprises of 224 amino acids and it sensitizes other organs to the effects of insulin. It kind of creates insulin resistance, and it protects against atherosclerosis. It inhibits inflammatory responses, and there are metabolic roles. Basically, synthesis actually blocks the energy consuming pathways and uh, upregulates the energy producing pathways. Okay, so fatty acid uptake by myocytes, rate of beta oxidation blocks fatty acid synthesis, it blocks neoglucogenesis. So all the pathways that require energy are blocked. All the pathways that creates energy, that uh, goes to those are anabolic pathways, they are upregulated, and it triggers the phosphorylation and activation of AMPK, that is AMP kinase. So AMPK mediated adiponectin action. So basically, uh, adiponectin activates this AMPK, and AMPK, as you know, it's a kinase. So
But will be activated by hormone sensitive lipases, Mama. There's another question in the assignment.
what is the question? Can you tell me what is the question?
And but it will be under the control of hormone sensitive lipases, no, ma'am.
It will be
There's one more question in the assignment.
What is the question in the assignment? Can you tell me?
It will be activated by hormone sensitive lipases.
A,
Adiponectin and leptin will be activated by their given list of
ah,
enzymes.
so
Pancreatic
yes,
lipases
yes, yes. So
and
adiponectin will be activated by hormone sensitive lipase. That's what you're saying, right?
yes, ma'am. One more question was there in that.
So basically, what I'm talking about is what will be the effect on the basic on the AMPK. So hormone sensitive
Your voice is
lipases
not
will
clear, ma'am. Your voice is getting break.
uh, just give me 1 minute.
Your voice is not clear, ma'am.
Yeah, just give me 1 minute. I'll just connect
Yes, ma'am.
my earphones. Hi, is it better?
Ah, yes, ma'am.
Yeah. So I'm saying, so hormone sensitive lipases, they activate different adipokines like adiponectin, leptin, okay? And then adiponectin in turn activates AMPK, okay? So here, so when adiponectin is activated, adiponectin will go and activate the AMPK, and AMPK being a kinase will go and phosphorylate different enzymes, and that phosphorylation will basically block all the energy consuming processes, and it will activate all the energy producing processes. So it will activate glycolysis, it will activate fatty acid oxidation. Okay. So, do you understand this point? Like what hormone
Yes,
sensitive
ma'am.
lipase is doing? Huh? So here you can see in this chart all the energy consuming processes like lipolysis, cholesterol synthesis, triacylglycerol synthesis. So all those pathways are blocked by AMPK, as well as glycolysis, fatty acid oxidation, glucose transport. All these pathways are activated by AMPK because these are energy producing processes. And adiponectin acts through its receptor in the brain to stimulate feeding behavior, and it inhibit the energy consuming physical activities, uh, and it regulates specific enzyme in key metabolic processes by phosphorylation. Okay, so since it's a kinase, I've already mentioned that. uh, it phosphorylates different enzymes, and depending on the enzyme, the enzyme either gets activated or inhibited. Is that clear to you?
Yes, ma'am.
Can you mute your mic? It will cause disturbance in the recording otherwise. Thank you. You can unmute when you have a question, okay? Don't worry. So, moving on to the next question now. Reduced levels of adiponectin in obesity are associated with which metabolic outcome? Option A, improved insulin sensitivity. Option B, increased glycogen breakdown. Option C, reduced gluconeogenesis. And option D, insulin resistance. Reduced level of adiponectin in obesity are associated with which metabolic outcome? Improved insulin sensitivity, increased glycogen breakdown, reduced gluconeogenesis, or insulin resistance. You're saying option D, that is insulin resistance. That is the correct answer. So, I've already mentioned that adiponectin activates the energy producing pathways and inhibits the energy consuming ones. So when adiponectin is low, in that case gluconeogenesis uh, and all the energy consuming processes will be upregulated, and there will be insulin resistance that will uh, be present due to insulin resistance also. Now you know that insulin also uh, activates the anabolic pathways, right? All the energy producing pathways. So insulin activates glycolysis and fatty acid oxidation, but when adiponectin is low, it will create insulin resistance. As a result, all the catabolic pathways will be upregulated and anabolic pathways will be downregulated. I hope that's clear. Moving to the next question then.
Yes, ma'am.
Which adipokine is primarily decreased in obesity and is protective against metabolic syndrome? Which adipokine is primarily decreased in obesity and is protective against metabolic syndrome? Option A leptin, option B adiponectin, option C TNF alpha, option D resistin. Which adipokine is primarily decreased in obesity and is protective against metabolic syndrome? Option A leptin, option B adiponectin, option C TNF alpha, and option D resistin. Which adipokine is primarily decreased in obesity and is protective against metabolic syndrome? Option A leptin, option B adiponectin, option C TNF alpha, and option D resistin. You are saying option A. We just discussed which adipokine is decreased during obesity. The previous question actually mentioned the answer. It's option B, adiponectin. So, adiponectin is decreased during obesity, and that reduced adiponectin in turn causes insulin sensitivity. So, the previous question was actually giving the answer to this question, reduced levels of adiponectin in obesity. Okay, so during obesity, insulin resistance is manifested, and which leads to changes in the activities of signaling enzymes and transcription factors, and adiponectin synthesis in adipocytes and adiponectin level in the blood decreases with obesity, which uh, and it is seen to be increased during weight loss. So as you know that adiponectin is uh, favoring all the anabolic processes and it is inhibiting all the catabolic processes. Okay, so all the energy consuming processes are inhibited. So during obesity, now you if you think logically, when a person is obese, that means more and more energy will be consumed in that body, right? So in that case, during obesity, logically all the factors that are going to uh, activate the anabolic processes, they are going to be down regulated. So insulin resistance is there, adiponectin will be reduced, because all those things are favoring the anabolic processes. All those things that are favoring the catabolic processes, they will be increased. So during obesity, uh, the adiponectin levels will be reduced so that the catabolic pathways will be upregulated. And when during weight loss, obviously anabolic pathways, because the body needs more and more energy. So in that case, energy producing uh, pathways will be upregulated, right? So if you think like that, you will understand. And obviously during obesity, there's chronic inflammation of adipose tissues. Is this clear to you? Why adiponectin is reduced during obesity? Okay. Moving to the next question. Leptin mainly acts on option A liver, option B adipose tissue, option C hypothalamus, option D skeletal muscle. Leptin mainly acts on option A liver, option B adipose tissue, option C hypothalamus, and option D skeletal muscle. You are saying option B adipose tissue. Actually, the correct answer is option C hypothalamus. Oh, wait, the correct answer would be option C, I think. I've made a typo here. So, leptin has receptors in hypothalamus. Uh, I'll correct this and upload the slides. So, leptin mainly acts on the hypothalamus of the brain. Uh, so, in the hypothalamus, there are receptors to which leptin can bind, and its principal role is in energy homeostasis via curtailing appetite and induces satiety. So it makes you feel full. So what it does is there are two types of nucleus, arcuate nucleus in the brain. One that makes you feel hungry, and that one that makes you feel full. So in simple words, leptin basically activates the anorexic neuron that makes you feel anorexic, or that induces satiety, that makes you feel full. So it curtails your appetite. And leptin stimulates the sympathetic nervous system, increasing blood pressure, heart rate, and thermogenesis by uncoupling. Okay. So leptin is released from the adipose tissue. It acts on the hypothalamus of the brain. Okay. So you can see here from the adipose tissue, it is released, and it will go and act on the hypothalamus of the brain, where it will release certain neuronal signals, and those neurons then uh, bind to the adrenergic receptors, and that will in turn uh, release a cascade of reactions, activate CAMP dependent protein kinase, and then eventually activate the different pathways. Okay. Is this clear?
Yes, it's secreted from adipose tissue, but it is
Yes.
regulated by hypothalamus.
It acts,
Okay.
so it is
Okay.
released from the adipose tissue, it goes and
One
acts
acts
on the hypothalamus.
hypothalamus.
So I haven't added this next slide to this. So if you see the lecture videos, so leptin basically binds to the receptor that is present on the hypothalamus, and that receptor goes and dimerizes, and it's a JAK-STAT pathway. It's a common cell signaling pathway. So you can go through the lecture video slides to understand the functioning of leptin that I have not added here. But then basically in general, in our body, any metabolite or any uh, hormone, all of these are ultimately going to interact with the cell signaling pathways. So there are different kinds. So like we have studied the CMP mediated protein kinase, right, where it is phosphorylating the protein kinase, and then protein kinase is going and phosphorylating different enzymes. So all these are called cell signaling pathways. So JAK-STAT is also one type of cell signaling pathway, and leptin actually binds to a receptor that has that particular uh, how do I say, particular compound, or particular cytokine to which basically signals the transcription factor, which basically activates a particular transcription factor that will finally activate or uh, basically upregulate or downregulate the pathways depending on its function. So this is a generalized process that happens in our body for different things. So in this case, since we are talking about leptin, leptin is also a cytokine, right? It's a adipokine, which is a type of cytokine, and these are the cytokines that are released from the adipose tissues are named as adipokines. So adiponectin, leptin, all these resistin, all these are different kinds of adipokines, and this leptin goes and signals different cell signaling pathway, which will ultimately activate or downregulate the pathways based on its role. Okay. Moving on to the next question. Most characteristic lipid abnormality in metabolic syndrome is option A, elevated LDL, option B, low HDL and high triglycerides, option C, low VLDL, and option D, high chylomicrons. Most characteristic lipid abnormality in metabolic syndrome is option A, elevated LDL, option B, low HDL and high triglycerides, option C, low VLDL, and option D, high chylomicrons. You're saying option B, low HDL and high triglycerides. That is the correct answer. So, basically, this is a characteristic of lipid abnormality, which is called dyslipidemia. So, obesity related dyslipidemia. It is primarily characterized by the increased level of plasma free fatty acids and triglycerides and decreased level of HDLs and abnormal LDLs, uh, and uncontrolled lipolysis is seen, due to which VLDL synthesis increases. So all the free fatty acids, there is a high amount of free fatty acids and triglycerides in the circulation in the blood, and that leads to uncontrolled lipolysis where VLDL formation increases, synthesis of VLDL increases, and this again, because of the synthesis of VLDL, increased levels of free fatty acids can uh, uh, synthesis of VLDL can actually cause impaired lipolysis, and it can actually increase the concentrations of free fatty acids in the circulation and decrease the concentrations of chylomicrons. So, uh, and these free fatty acids in the circulation, it can decrease the mRNA expression or activity of lipoprotein lipase in the adipose tissue and skeletal muscle, and it inhibits the lipolysis of chylomicrons, promoting hypertriglyceridemia. So triglycerides and free fatty acids are increased in the plasma, increased in the blood, leading to the condition hypertriglyceridemia, because triglycerides are high in the blood. So hypertriglyceridemia. Okay. So the characteristic feature is uh, low HDL and LDL, and high triglycerides. Okay. So hypertriglyceridemia, it further triggers the cholesterol ester transfer protein mediated exchange of triglycerides for cholesterol esters between the triglyceride rich lipoproteins, that are VLDL and IDL, and lipoproteins which are relatively richer in cholesterol esters. So from VLDL and IDL, the triglyceride exchange to LDL and HDL, uh, it uh, it is triggered, and this leads to decreased HDL cholesterol concentration and a reduction in the triglyceride content in the LDL as well. Because so basically you understand, free triglycerides are present more, and the triglyceride concentration from the LDL will decrease, cholesterol concentration from the HDL will decrease, and increased triglycerides in the LDL is hydrolyzed by hepatic lipase, leading to the formation of the small dense LDL, which causes high risk of cardiovascular diseases. Okay, I hope this is clear.
Yes, ma'am.
Moving to the next question. Ethanol metabolism mainly generates acetaldehyde via which enzyme? Ethanol metabolism mainly generates acetaldehyde via which enzyme? Option A, aldehyde dehydrogenase. Option B, alcohol dehydrogenase. Option C, catalase. Option D, monoamine oxidase. Ethanol metabolism mainly generates acetaldehyde via which enzyme? Option A, aldehyde dehydrogenase. Option B, alcohol dehydrogenase. Option C, catalase. And option D, monoamine oxidase. You are saying option B, that is alcohol dehydrogenase, that is the correct answer. So, ethanol is metabolized in the liver by the liver alcohol dehydrogenase. It is a cytosolic enzyme. It is present in the cytosol of the liver cells, and uh, ethanol is converted into acetaldehyde. Acetaldehyde is then converted into acetate by uh, acetaldehyde dehydrogenase, which is a mitochondrial enzyme. So basically, ethanol is converted into acetaldehyde, and then acetaldehyde goes into the mitochondria, where acetaldehyde dehydrogenase converts it into acetate, and this acetate can then uh, be converted into acetyl coenzyme A for the acetyl coenzyme A for which can enter the TCA cycle, or which can uh, uh, go in the fatty acid synthesis depending on its fate. Okay, so during ethanol consumption, the ethanol goes into the liver. It is metabolized in the liver by the enzyme alcohol dehydrogenase. And as I've mentioned in the previous sessions also, when we see the enzyme dehydrogenase is there, that means involvement of NAD is there. There's a there's one molecule of NAD getting converted into NADH plus H plus with this alcohol dehydrogenase, as well as acetaldehyde dehydrogenase. So if you see here, two with uh, in both the steps, one NADH is forming. Okay, and now I have mentioned that this step is happening in the cytosol, whereas the second step is happening in the mitochondria. So that means the cytosolic as well as the mitochondrial NADH concentration is going to increase. Okay, I hope that's clear. Moving to the next question. In alcoholic liver disease, the increased NADH by NAD plus ratio inhibits which major process? In alcoholic liver disease, the increased NADH by NAD plus ratio inhibits which major process? Option A, beta oxidation. Option B, glycolysis. Option C, gluconeogenesis. Option D, glycogenolysis. In alcoholic liver disease, the increased NADH by NAD plus ratio inhibits which major process? Option A, beta oxidation. Option B, glycolysis. Option C, gluconeogenesis. And option D, glycogenolysis. You have answered option B, that is glycolysis. Uh, Actually, that's the wrong answer. The correct answer is option A, beta oxidation. So, uh, during the uh, alcohol, like chronic alcohol consumption, chronic alcohol use, the alcohol, as I've mentioned, it is metabolized in the liver. And I've told you that it is metabolized by the alcohol dehydrogenase to convert it into acetaldehyde, and then acetaldehyde gets converted into acetate by acetaldehyde dehydrogenase. And in both cases, the NADH, in one case, the NADH is secreted in the cytosol, in the second enzyme, the NADH is being secreted in the mitochondria. So you see the concentration of NADH is increasing both in cytosol and mitochondria of the liver. So overall ratio of NADH by NAD will be very high, right? Now, NADH has a inhibitory effect on the beta oxidation pathway. Why? Because if you see the beta oxidation pathway produces what? NADH and FADH2, right? If you remember, recall the beta oxidation pathway where the activated fatty acids are uh, broken down into two carbon product, that is acetyl coenzyme A, and any high amount of NADH, because after every step NADH is being released. So a lot, if we have a long chain fatty acid, when it is catabolized by beta oxidation, a lot of NADH is being released from the fatty acid oxidation, right? So now beta oxidation is triggered when the mitochondrial NADH concentration goes down, when the cell senses that we need more NADH for the TCA cycle. And you know that NADH is where the NADH is going. NADH is going to the electron transport chain, right? Here the electron transport chain is shown. NADH is going to the electron transport chain to make ATP, that is the energy currency. So when the cell requires energy, it breaks down the fatty acids by beta oxidation to produce more and more NADH, so that it can go and enter the electron transport chain and make more and more ATP, that is energy. That's how we get the energy from the fatty acid pathway, right? Now, if the cell is already sensing that there is enough NADH, cell will think that, okay, we don't need any more energy. We have enough NADH, and NADH will have inhibitory effect on the beta oxidation pathway. Okay. NADH actually does not have any inhibition effect on glycolysis. It will have the
But it will go for anaerobic glycolysis, no, ma'am, for lactate production in case of that will be the effect of reversal of NAD to NADH on
yes,
glycolysis.
yes, but that's a pathway that is going separately in the cytosol, right?
Okay.
This what we are talking about here, the NAD to NADH ratio, high NAD to NADH ratio, sorry, high NADH to NAD ratio, it is not forcing glucose to go towards lactate, right? So more and more the pyruvate, instead of entering the TCA, it goes towards the lactate, where the NADH is being utilized, the cytosolic NADH is being utilized. But majorly the NADH that is the high NADH amount, majorly it goes and inhibits the beta oxidation pathway. This type of inhibition is a feedback inhibition. Okay, so NADH is a product of beta oxidation pathway. So NADH has a feedback inhibitory effect on the uh, enzyme that starts the beta oxidation of the activated fatty acids. Okay, So there will be accumulation of the activated fatty acid, that is fatty acyl coenzyme A. Is this clear to you?
Yes, ma'am. It's beta oxidation that is getting inhibited, but glycolysis there will be uh,
Glycolysis, yes. So
it will be anaerobic
so
path.
yeah, glycolysis is not inhibited here, right?
Okay.
Glycolysis is happening. Glycolysis is glucose to pyruvate. This part is only glycolysis, right? From glucose
Okay,
to
ma'am.
pyruvate, that is glycolysis. After that, pyruvate has different fate. Now,
So
pyruvate
beta
can go
oxidation is that
towards,
one which is getting inhibited.
yes, beta
Okay,
oxidation
ma'am.
is the pathway that is being inhibited. Glycolysis is not inhibited here. Glycolysis is
Okay, okay,
happening
yes.
because we are
Okay, ma'am.
getting the pyruvate formation. After
Okay, ma'am.
pyruvate, instead of pyruvate going to acetyl coenzyme, pyruvate goes towards
For lactate production.
lactate production. Yes.
Okay, ma'am. It's clear, ma'am.
Okay. So in the question, it was asked which process is inhibited. So that is beta oxidation. Moving to the next question then. In hepatic cells, NADH accumulation due to ethanol metabolism primarily leads to option A, enhanced gluconeogenesis, option B, ketone body synthesis inhibition, option C, fat accumulation, that is steatosis, option D, activation of TCA cycle. In hepatic cells, NADH accumulation due to ethanol metabolism primarily leads to enhanced gluconeogenesis, ketone body synthesis, fat accumulation, that is steatosis, and activation of TCA cycle. What would be the correct answer? Yes, option C is the correct answer. Fat accumulation or steatosis. So, I have already told you that beta oxidation is inhibited by the high amount of NADH that is there. Beta oxidation is being inhibited. Now, fatty acids are getting activated and making fatty acyl coenzyme, right? So now fatty acyl coenzyme cannot go in this direction towards acetyl coenzyme, but so it will instead go towards the triacyl glycerol formation, which will lead to VLDL synthesis, which will be basically high amounts of lipids in the circulation, as well as these triacyl glycerol and VLDL will enter the liver, and lipids will get deposited in the liver, leading to fatty liver or hepatic steatosis. Okay, so basically fat accumulation occurs due to high NADH secretion during alcohol metabolism. Okay, I hope this is clear. This is related to the previous explanation only. Moving to the next question. The microsomal ethanol oxidizing system, that is MEOS, mainly involves option A, cytochrome P450 2E1, option B, alcohol dehydrogenase, option C, catalase, option D, NADH dehydrogenase. The microsomal ethanol oxidizing system, that is MEOS, mainly involves option A, cytochrome P450 2E1, option B, alcohol dehydrogenase, option C, catalase, and option D, NADH dehydrogenase. Yes, you are right. Option A is the correct answer, cytochrome P450. So this is another alcohol metabolic pathway, along with the alcohol dehydrogenase. One type of cytochrome P450, that is the 2E1 variant. So cytochrome P450 CYP2E1, it is a mixed function oxidase. It can also perform the oxidation of ethanol into acetaldehyde, and this enzyme actually uses NADPH. So you see, in case of alcohol dehydrogenase, NAD was getting converted into NADH, whereas in case of cytochrome P450, ethanol is being oxidized and NADPH is being used. Okay, not NADH, NADPH uh, will be used. It will act as the electron donor. So the electron donors are ethanol and NADPH, by which oxygen is reduced to water. So you see here, oxygen is the electron acceptor. NADPH and ethanol will donate the electron, and oxygen is reduced to water. NADPH is oxidized to NADP plus, and ethanol is oxidized to acetaldehyde. Okay, and this route accounts for only 10 to 20% of the ethanol oxidation. Rest of the ethanol is obviously oxidized in the liver by metabolized in the liver by alcohol dehydrogenase. Okay, so this occurs in the microsomes, liver microsomes. This oxidation occurs in the liver microsome by the enzyme cytochrome P452E1. It belongs to the cytochrome P450 family. Okay, I hope that's clear. Moving to the next question. Most toxic metabolite in alcohol metabolism is. The most toxic metabolite in alcohol metabolism is option A acetate, option B acetaldehyde, option C ethanol, option D pyruvate. The most toxic metabolite in alcohol metabolism is option A acetate, option B acetaldehyde, option C ethanol, and option D pyruvate. You have answered option B, that is acetaldehyde. That is the correct answer. So acetaldehyde causes the major toxicity during the ethanol abuse. So chronic ethanol abuse, uh, it can actually manifest fatty liver, alcohol induced hepatitis, or cirrhosis, depending on the severity. And you know that ethanol is oxidized into acetaldehyde. Acetaldehyde accumulates in the liver and it is released into the blood after heavy doses of ethanol. And because ethanol is converted into acetaldehyde, then acetaldehyde is converted into acetate by the mitochondrial acetaldehyde dehydrogenase. Okay, now if there's a high consumption of ethanol, more and more acetaldehyde will get accumulated. Not all the acetaldehyde can be converted into acetate that for entering into TCA cycle eventually by acetyl coenzyme A. Then in that case, what will happen? The acetaldehyde that is not being converted into acetate, they will be released into the blood, and acetaldehyde is highly reactive, so it can go and bind to amino groups, sulfhydryl groups, nucleotides, phospholipids to form adducts, and these adducts are toxic to the body. Okay, so how it causes alcohol induced hepatitis? It generally decreases the hepatic protein synthesis like calmodulin synthesis, ribonuclease, tubulin, and it can also decrease the protein synthesis in the heart and other tissues. Acetaldehyde adducts of tubulin actually diminishes the secretion of serum proteins and VLDL lipoproteins, and accumulation of serum albumin, coagulation factor, transport proteins for vitamins, steroids, and iron in the liver together with lipid. All these are manifested by acetaldehyde. Okay, so the acetaldehyde, uh, the toxic effect of acetaldehyde actually affects the uh, secretion of serum proteins. It affects the secretion of VLDL, and accumulation of certain proteins results in an influx of water within the hepatocytes and a swelling of liver that contributes to the portal hypertension and disruption of hepatic architecture occurs. So, uh, acetaldehyde can actually lead to alcohol induced hepatitis. It can also induce free radical damage. So, I told you that the adducts that are forming by acetaldehyde, it can bind. So, one of the adduct that can form is by binding to glutathione. Now, glutathione, if you remember from the previous session, it's a tripeptide, right? And it has multiple roles uh, in protecting us against free radical damage. So, acetaldehyde binds to glutathione and then diminishes its ability to protect against the free radical mediated damage. So, with chronic consumption of ethanol, mitochondria becomes damaged, which affects the rate of electron transport and oxidative phosphorylation, eventually decreasing the amount of ATP being produced. Then fatty acid oxidation is also decreased even further because NADH will keep on increasing, thereby enhancing lipid accumulation, which can lead to fatty liver, as we have seen earlier. Mitochondrial changes further impair mitochondrial acetaldehyde oxidation, thereby initiating a cycle of progressively increasing acetaldehyde damage. So, you see how it is a cyclical process. So, if we have consumed more ethanol, more acetaldehyde will form. It is going to damage the mitochondria where the acetaldehyde can be converted into acetate. Now, because the mitochondria is being damaged, more and more acetaldehyde accumulation will occur, and it will actually amplify the acetaldehyde uh, dependent damages in the cell. So, more free radical damage will occur, more and more hepatitis will be induced, and all those toxic conditions will be occurring. So this is then how free radical formation occurs due to ethanol uh, by the second method. So I told you that ethanol is also metabolized by the cytochrome P450 pathway. So increased production of free radical can also occur by that pathway. FAD and FMN in the reductase and the heme in the cytochrome P450 system, uh, they can be they can affect uh, and hydroxyethyl radical forms during this ethanol metabolism by cytochrome P450. So that's another pathway which can eventually activate the carcinogens in the liver, ultimately leading to liver cancer. Okay, I hope you understand how acetaldehyde is toxic to the cell, how it is forming the adducts. Main point is to remember that acetaldehyde, when accumulated, it is released into the blood and it can react to multiple groups and it can form adducts with different biomolecules, and all those adducts are actually very toxic. They can uh, induce free radical damage, they can induce hepatitis. Okay, is this clear? Okay, I'll move on to the next question then, if you don't have any doubt. Respiratory quotient of carbohydrate oxidation is option A 0.7, option B 0.8, option C 1, option D 1.2. Respiratory quotient for carbohydrate oxidation is. Do you have any doubt in the previous question? Okay. Okay, so you can answer this question. Respiratory quotient for carbohydrate oxidation is option A 0.7, option B 0.8, option C 1, option D 1.2. What would be the correct answer? Do you know the correct answer? So the correct answer to this question is option C 1. The respiratory quotient, it is a measure of the ratio between the volume of CO2 produced and the oxygen consumed. So RQ is equal to CO2 eliminated divided by the oxygen that is consumed. So carbohydrates, they have a RQ of 1, indicating that for each gram of oxygen consumed, 1 gram of CO2 is produced. Fats have a RQ of 0.7 and proteins have a RQ of 0.8. And in a mixed diet, RQ typically averages from 0.82 to 0.85. So these are just some numbers you should remember. It can come as an MCQ question. That's why I've added this. When the utilization of fats increases relative to carbohydrates, the RQ decreases in a mixed diet, right? Because fats have a lower RQ and carbohydrates have a higher RQ. So this decreases. This decrease in the RQ is observed in diabetes mellitus, where carbohydrate utilization is reduced, uh, which is lowest during the active ketolysis. Oxygen consumption is measured using Benedict Roth apparatus, which is shown here. So again, this could be a multiple choice question, so you should remember that. Moving on to the next question. Yes. Do you have any question? You are raising your hand. Do you have any question? Am I not audible? If you have any doubt, please unmute and ask. Okay, otherwise I'll move on to the next question. Question number 16. A person suffering from protein energy malnutrition shows which of the following biochemical features? A person suffering from protein energy malnutrition which of shows which of the following biochemical features? Option A, hypoglycemia. Option B, albuminemia. Option C, hyperglycemia. And option D, both A and B. A person suffering from protein energy malnutrition shows which of the following biochemical features? Option A, hypoglycemia. Option B, hypoalbuminemia. Option C, hyperglycemia. And option D, both A and B. What do you think would be the correct answer? So the correct answer is option D, that is both hypoglycemia and hypoalbuminemia. So during protein energy malnutrition, multiple biochemical features are present. The metabolic rate is decreased. Albuminemia is seen, especially during kwashiorkor. Uh, Retinol binding protein is very low. IgG increases due to associated infection. Fatty liver may be associated in kwashiorkor, but not in marasmus. And it is due to decreased lipoprotein synthesis and decreased availability of DLDL. The serum free fatty acids is usually high. Glucose tolerance is often normal. Hypoglycemia may be seen in marasmic child, and hypokalemia is and dehydration can be seen if there is diarrhea. Hypomagnesemia may be seen. So basically, uh, you know the terms hypoglycemia, hypomagnesemia. So basically, hypo means reduced, glycemia means presence of glucose in the blood, magnesemia means presence of magnesium in the blood, albuminemia means presence of albumin protein, serum albumin in the blood. So if hypo is there, that means the albumin levels are low. Glycemia, hypoglycemia, glucose levels are low in the blood. Okay, and total body water content increases in kwashiorkor up to 60% of the weight. Okay, do you have any doubt? Moving on to the next question then. Kwashiorkor is characterized by option A, wasting without edema, severe protein deficiency with edema, deficiency of both energy and protein, option D, vitamin deficiency only. Kwashiorkor is characterized by Kwashiorkor is characterized by option A, wasting without edema, sorry. Option B, severe protein deficiency with edema. Option C, deficiency of both energy and protein. Option D, vitamin deficiency only. So the correct answer to this question is severe protein deficiency with edema. So in protein energy malnutrition, that is PEM, it is sometimes called protein energy undernutrition also. It is a form of malnutrition that is defined as a range of condition arising from coincident lack of dietary protein or energy in varying proportions. So there are different types of uh, protein energy malnutrition. One where protein deficiency is there, one where is major energy deficiency is there. So kwashiorkor is the variant where protein malnutrition is predominant. Marasmus is the variant where calorie intake is def- mm, deficiency in calorie intake is predominant. Marasmic kwashiorkor, there is a variant called marasmic kwashiorkor which is marked by protein deficiency and calorie deficiency together, and it is sometimes referred to as the most severe form of malnutrition. So kwashiorkor, it is characterized by protein deficiency. Marasmus is characterized by calorie deficiency, and marasmic kwashiorkor is characterized by both protein and calorie deficiency. Okay. If you have any doubt, let me know. Moving to the next question. Marasmus differs from kwashiorkor by having option A, edema, option B, fatty liver, option C, no edema and severe wasting, option D, high plasma properties. Marasmus differs from kwashiorkor by having option A, edema, option B, fatty liver, option C, no edema and severe wasting, and option D, high plasma protein. Is there any problem with your connection? Can you hear me?
Actually, I was not able to see your slides, ma'am, for quite some time. There's network issue, I think.
Okay,
But,
yeah.
but there is no chat box for me to write the answers. I'm not getting that option.
Still
Uh,
not getting the option for chat box.
no, yes, ma'am. I don't know what to do.
Okay, you can unmute and answer, no problem.
Yes, ma'am.
Do you want me to show the previous slides?
Yes, ma'am. 17 and 16 questions I didn't see, ma'am.
You saw the question number 15, right? Respiratory quotient.
No, ma'am. From 15, respiratory question for carbohydrate oxidation.
Respiratory quotient for carbohydrate oxidation is. So, do you know the answer?
Mm, 0.7.8 0. Is it option C, ma'am?
Yes. Option C is the correct answer. So, respiratory quotient is basically the amount of or the volume of CO2 produced divided by the amount of oxygen consumed. Okay, so for every nutrient, like carbohydrate has a respiratory quotient of 1. That means each gram of oxygen consumed, 1 gram of CO2 is produced. For fats, the respiratory quotient is zero point 0.7. That means uh, the amount of uh, CO2 eliminated, it is less than the amount of oxygen consumed. For proteins, the RQ is zero point 0.8. In a mixed diet, the RQ typically averages from 0.82 to 0.85. So that means if you increase the fat in your diet relative to carbohydrate, your RQ will decrease because carbohydrates have a higher RQ, right? So that will overall decrease the RQ for the overall diet. So that is, so it's just a measure of how much CO2 is eliminated per gram of oxygen that is consumed. Okay. Then the next question was person suffering from protein energy malnutrition shows which of the following biochemical features.
Hypoglycemia, hyperglycemia. It's both A and B, mama.
Yes. So uh, in protein energy malnutrition, hypoalbuminemia and hypoglycemia, these are some common biochemical features along with hypokalemia and hypomagnesemia. Okay. Then the next question was kwashiorkor is characterized by option A wasting without edema, option B severe protein deficiency with edema, option C deficiency of both energy and protein, option D vitamin deficiency of both.
It's severe protein deficiency with edema.
Huh. So kwashiorkor. So there are different types of protein energy malnutrition. So either protein can be deficient or energy can be deficient or both can be deficient. So in case of protein malnutrition, kwashiorkor, like kwashiorkor is the type where protein malnutrition is predominant. Marasmus is another type of protein energy malnutrition where calorie intake is deficient. So when the person is deficient in calorie intake, it leads to marasmus type. And there is third variant which is marasmic kwashiorkor, which marks the protein and the calorie deficiency together. Okay. Then I was on this question. Marasmus differs from kwashiorkor by having option A edema, option B fatty liver, option C no edema and severe wasting, and option D high plasma protein.
There's no edema and there will be severe wasting.
Yes. So the major difference between marasmus and kwashiorkor, I told you, kwashiorkor is characterized by the protein deficiency, whereas marasmus is characterized by the calorie deficiency. And if you see the age of onset, marasmus is uh, the age of onset for marasmus is less than 1 year, whereas kwashiorkor occurs later in during the 1 to 5 year stage. Marasmus occurs due to early weaning and repeated infections, whereas kwashiorkor is due to excessive starchy diet, basically lack of protein in the diet uh, causes kwashiorkor. Uh, then growth retardation, it is marked in marasmus and it is present in kwashiorkor. The attitude of the kid becomes irritable and fretful during marasmus, whereas in kwashiorkor the kid becomes lethargic and apathetic. Then there is shrunk appearance becomes very shrunken in case of marasmus because it's a calorie deficient malnutrition type. The person becomes dehydrated, whereas in kwashiorkor the person looks plump due to edema on face due to accumulation of fluids in the tissue. So edema is basically liquid accumulation. Lower limb shows also show edema. So basically, the body looks swollen due to fluid accumulation. It's not fats that are accumulating, it's due to the edema. The body looks plump. Okay. And the appetite in marasmus is normal, but in kwashiorkor the person becomes anorexic, where they feel full and they cannot eat more. Okay. And then the skin looks dry and atrophic in marasmus. And in kwashiorkor, crazy pavement dermatitis occurs due to peeling, cracking, and denudation. Then hair, there's no characteristic change in the hair in marasmus, but in kwashiorkor, sparse, soft and thin hair, curls may be lost. Then there are some other associated features. You can read these in the slides as well. Okay. Moving to the next question. Insoluble fibers mainly help in option A, lowering blood sugar, option B, reducing LDL cholesterol, option C, increasing bowel movement, and option D, providing calories.
Increasing bowel movements.
Yes. So insoluble fibers, mainly like cellulose, hemicellulose, they retain water and they increase increase the bowel movement. So it's part of a dietary fiber. So there are different types of dietary fiber. Insoluble fibers are known to increase the bowel movement. Some soluble fibers are actually known to reduce the cholesterol. So there are different features of dietary fibers. Next question. Cancer cells primarily depend on option A, oxidative phosphorylation, option B, aerobic glycolysis, option C, fatty acid oxidation, and option D, gluconeogenesis. Cancer cell primarily depend on oxidative phosphorylation, aerobic glycolysis, fatty acid oxidation, and gluconeogenesis.
It's aerobic glycolysis, no, ma'am.
Yes. So unlike normal cells, cancer cells actually depends on aerobic glycolysis for its energy demand. Normal cells undergo oxidative phosphorylation for the ATP production, but cancer cells. So this was found by Warburg, and the effect is called Warburg effect. Because earlier it used to be assumed that the cancer cells are getting the energy by the same way, but later it was found that the cancer cells, instead of going towards the oxidative phosphorylation towards the electron transport chain, the pyruvate actually makes lactate, which is less uh, uh, basically it is easier for the cells to produce more and more metabolite in a shorter time and proliferate more, because now cancer cells are basically the cells that are proliferating without any control, right? Without any check or without any regulation. So they are continuously dividing. So they need to produce quick source of energy. They cannot rely on a long process of electron transport chain oxidative phosphorylation for the ATP production. They have to produce the metabolites as quickly as possible so that they can divide more and more in a very short time. Okay. So in that case, instead of going towards the oxidative phosphorylation, they go towards the lactate formation, even in the presence of. So we have studied that lactate formation occurs from pyruvate in the absence of oxygen, right? But cancer cells have a way to produce lactate from pyruvate even in the presence of oxygen. Okay, that's why it's called aerobic glycolysis, which is leading to lactate formation. Is that clear?
Yes, ma'am.
Next question is the Warburg effect provides cancer cells with option A, more ATP, option B, more oxygen, option C, low NAD plus regeneration, and option D, biosynthetic intermediates for growth. Warburg effect provides cancer cells with option A, more ATP, option B, more oxygen, option C, lower NAD plus regeneration, and option D, biosynthetic intermediates for growth.
Is it lower NAD regeneration, ma'am? Because oxygen that has to enter into ATC.
No, actually, it is biosynthetic intermediates for growth. So, I told you, cancer cells basically have one goal. They just want to divide continuously and proliferate throughout the system. Okay, they are cancerous, right? The meaning of the cell being cancerous is that it is dividing continuously, it is growing. That's why we get those tumors during cancer, because the cells have no checkpoint, no regulation. They are continuously dividing and multiplying in number and increasing in number. That's how they spread throughout the body also. Okay, so the goal of a cancer cell is very simple. They just want to grow. Okay, so that's why instead of going through the conventional oxidative phosphorylation, they go through this Warburg effect, which is basically aerobic glycolysis, where pyruvate is forming lactate instead of acetyl coenzyme and the TCA cycle. It is forming lactate. This is actually giving the cell more and more intermediates for the growth. So, glycolysis is continuously happening. The cell is completely dependent on glycolysis for energy. Okay, from glycolysis, whatever ATP it is getting, it is depending on that ATP, and it is dependent on the intermediates of the glycolysis. So, the different intermediates that are formed during glycolysis. I haven't added. So basically, like glucose 6 phosphate, glycolysis starts from glucose, right? So glucose will form glucose 6 phosphate. Now you know glucose 6 phosphate can enter the HMP shunt pathway. So it will depend on the HMP shunt pathway from the glucose 6 phosphate. Then again, DHAP can enter some other pathway. So, like that, different intermediates can be used for the growth of the cell. The cell does not want to do anything else. It does not have to do any other, all the other metabolic pathways that you have studied. It just wants to grow continuously. It just wants to divide continuously. For dividing continuously, it needs uh, things to make protein. It needs things to make uh, DNA. So HMP shunt pathway is required for making the ribose, for making the NADPH. So all those things are met by the glycolysis pathway. So it solely depends on the glycolysis pathway, so that it can use all the intermediates uh, for its growth. Okay. So in a normal cell, in a mammalian cellular proliferation, there are extracellular stimulus such as growth factors, which interacts with the corresponding receptors to promote intracellular signaling cascades, and it is a finely controlled process that depends on the neighbor cells, chemical signals in the microenvironment, and cell itself. Whereas in cancer cells, there is no such controlled process or anything going on. Okay, the genetic mutations increase the number of growth factor receptors. Intracellular signaling pathways that promote the cell proliferation may be constantly turned on. So, in case of normal cell proliferation, in case of mammalian cell proliferation, the signaling pathways are tightly regulated. So, the when the cell sees that proliferation is required, only then those particular signaling pathways are switched on. When the cell sees that the cell needs to be killed, apoptotic pathways will be switched on. But in case of cancer cells, only proliferation is happening. Okay, so all the proliferation related pathways will be constantly on, and it will be amplified. And to support this continuous cell proliferation, biosynthetic capabilities of the tumor cells are increased, including fatty acid, nucleotide synthesis, metabolic uh, autonomy, and the advantages of Warburg effect to these cancer cells are, it is a short duration cell generates more ATP per unit time. So if you see the number of ATP generated through oxidative phosphorylation is obviously more than the number of ATP generated just by glycolysis. But if you see time wise, the in, let's say, in a minute the number of ATPs that can be produced only by glycolysis, if the cell is continuously just doing glycolysis, from the glucose it is making pyruvate and lactate, again from glucose it is making pyruvate and lactate. From that process itself, it can generate actually more ATP in 1 minute than the cell going through the whole glucose to pyruvate, then pyruvate to acetyl coenzyme, then the TCA cycle, then the oxidative phosphorylation. So it's a long process, right? So instead of that long process, the cancer cells are smart enough to choose a shorter process where it can quickly generate more ATP in a shorter time. Okay, and it is getting all the glycolytic intermediates, which are important sources of different biosynthetic pathways. Like I have told you, glucose 6 phosphate enters the pentose phosphate pathway to generate NADPH and nucleotide. So it will generate the ribose, which will go for the nucleotide synthesis. Shunting of 3 phosphoglycerate into serine and glycine biosynthesis pathway. So your protein, uh, for the amino acid metabolism for protein is also being taken care of. NADPH will go for the lipids biosynthesis. Phospholipid biosynthesis is enabled by the conversion of DHAP to glycerol 3 phosphate. So you see, all the intermediates of glycolysis can actually take care of the basic cellular needs that are required for the cell to divide and grow. So only that much is required, that much can be taken care of by glycolysis, and it can actually do the whole energy, like it can do the whole process in a shorter time than the long energy consuming process. Okay, then fructose 6 phosphate enters hexosamine pathway to support protein post translational modification, glutathione production, increased cancer cell survival and resistance to chemo and radiotherapy. Okay, is this clear to you why glycolysis is aerobic glycolysis is favored in case of cancer cells?
Yes, ma'am. It's clear, ma'am.
Moving to the next question now. A key enzyme upregulated in cancer glycolysis is option A pyruvate kinase M2, option B glucose 6 phosphate, option C citrate synthase, option D acetyl coenzyme carboxylase. A key enzyme that is upregulated in the cancer glycolysis is. Do you know the answer? Which enzyme is upregulated in cancer glycolysis? You're saying option A pyruvate kinase M2. Yes, that is the correct answer. So glucose transporters are overexpressed because aerobic glycolysis is happening, so you need more and more glucose. Then glycolytic enzymes are overexpressed, so hexokinase 1 and 2, triose phosphate isomerase 1, phosphoglycerate kinase 1, enolase A, pyruvate kinase M2, aldolase A, phosphoglucomutase, and pyruvate DHA1. All these enzymes are overexpressed, all these enzymes are upregulated. Activation of regulatory bifunctional enzyme 6 phosphofructo 2 kinase or fructose 2 6 bisphosphatase. uh, is also seen during aerobic glycolysis of cancer cells. Okay. Okay. The next question is lactic acid accumulation in tumor contributes to option A, alkaline microenvironment, option B, acidic microenvironment promoting invasion, option C, enhanced oxidative metabolism, and option D, increased oxygen tension. Lactic acid accumulation in tumor contributes to option A, alkaline microenvironment, option B, acidic microenvironment promoting invasion, option C, enhanced oxidative metabolism, and option D, increased oxygen tension. You are saying option B, that is acidic microenvironment promoting invasion, that is the correct answer. So lactic acid, you know that now you know that in the cancer cells aerobic glycolysis leading to lactic acid formation, and more lactic acid will obviously create an acidic environment leading to acidosis, and when tumor cells enhances glycolysis produces large quantities of lactic acid, they generate an environment that is more toxic to the adjacent normal cells than to the malignant cells. CO2 is a significant source of acidic extracellular pH in the tumor microenvironment. Acidosis also promotes extracellular matrix degradation, increasing invasiveness. So, basically, along with uh, continuously proliferating and dividing and growing, it is also being invasive to the neighboring normal cells. So all the normal cells surrounding the tumor cells, they are being intoxicated by the acidic environment of the tumor cells. So under normoxic acidosis, tumor cells inhibit proteasomal degradation of HIF1 via the nuclear sequestration of VHL HIF1, and some intracellular pH regulating systems that help cells cope up with the acidosis are sodium proton exchangers. Then you have monocarboxylate transporters, proton pumps. So all these are basically ways of the body to recover from the acidosis. But then when tumors keep on proliferating and keep on making lactic acid, after a point it becomes very invasive for the normal cells. It becomes toxic to the normal cells. The acidic environment of the tumor cells become toxic to the normal adjacent normal cells. Okay, so this was the last question of this session and of this course as well. uh, do you have any question, any form of doubt in any of the questions discussed today, or anything you want to ask?
We'll go through the questions once again, ma'am, for
uh,
get clear view.
You want me to go through all the
No,
questions?
no, no. I need to go through it once again, ma'am.
Okay, okay. So, yeah, you can go through. I have already uploaded the questions that I have discussed, and
Okay,
you have the questions that are there in the assignment for like for the course.
okay.
So, go through all the questions, go through the lecture slides. I think that should help you with the examination part.
Yes, ma'am.
Yeah. So this then, if you don't have any doubt, I think I can end the session. So this is the last session. So if you want to ask anything, you can ask now. Otherwise, all the best for your exam.
Thank you, ma'am. Thank you so
Yeah.
much for your very elaborative sessions all through weeks. It will be very helpful for our classes also, regular classes also.
Yeah. So
We
you
had
can,
an in-depth
yes,
discussion on the
yes,
questions.
yes, yes.
And we
You
have
can,
we have to.
yeah, you can go through the videos, recorded videos again if you have any doubts. So
Yes, ma'am.
mostly
Sure,
I've
ma'am.
tried to cover all the topics. So I hope that's helpful to you.
Yes,
Uh,
ma'am.
Prepare well for your exams.
Thank you so
Success,
much.
You are a master's student or?
I'm assistant professor, ma'am,
Oh, assistant professor where?
and Karaikal, Vinayaga Missions Medical College.
Oh, okay, okay. Then this course is actually helpful for you, more
Yes,
helpful.
ma'am. Very clear view of all the. Though we are teaching regularly,
Yeah,
some few aspects now only we are in clear view. So we have to go through it again so that it will be helpful for our regular classes.
this will help you get a content for your course preparation also.
Yes, ma'am.
Good. Okay, then I'll,
Thank you.
yeah, I'll end the session. Thank you so much
Okay.
for joining all the sessions.
Thank
Not
you,
many
ma'am.
students are joining.
Yes,
Yeah,
I used to join this properly in spite of all the other duties because it's very helpful. We can get a clear view. Though we are listening to the videos, lectures, this
yeah,
live session, the sessions are very helpful, ma'am. Thank you so
yeah,
much
yeah.
for
Thank
your
you.
time.
Yeah.
Thank you, ma'am.
Okay, then. Bye bye. All the best for your
Bye,
exams.
ma'am. Take care, ma'am.